Hello, everybody. Welcome to Online Learning with Miss E. I'm so excited you've clicked on this video and are going to do some online learning with me. This is the first video of 3 where we are going to talk about the central dogma of molecular biology. So the first thing we have to do is we have to break down this term. What does the central dogma of molecular biology mean? So, a dogma is just an accepted truth, something we accept to be true. And molecular biology is the study of biology at the molecular level, which means that we are talking about molecules like DNA, RNA, and proteins. So if we put these two terms together, then the central dogma of molecular biology is the explanation that scientists have accepted as true for how the traits we inherit in our DNA are able to actually be expressed at the molecular level. And the explanation scientists came up with for how we go from our inherited traits to our expression of those traits is this two-step process: DNA to RNA, and then RNA to protein. And this two-step process is what we call the central dogma of molecular biology. And that's what we're going to be talking about in the next 3 videos. So for today, before we get into that two-step process, we are just going to review DNA because that is where it all begins. So it is helpful to have a basic understanding of the structure of DNA and how it works. So we have discussed in this class that DNA carries the instructions for life. Our DNA is housed in our nucleus, the nucleus of all of our cells, and it is the genetic material that tells our bodies what to do, when to do it, and how. But DNA is just the instructions, and without a finished product, instructions don't do us any good. It's like having a recipe for a cake, but never getting the actual cake. So how do we go from the instructions to the proteins that we want? Well, this is where the central dogma of biology, of molecular biology, comes in. This process, DNA to RNA, and then RNA to protein, explains how we go from the DNA in our nucleus to the actual proteins we need to function. It explains how the traits we inherit are expressed. So for today, we're just going to talk about DNA. What is it? DNA is a nucleic acid that is composed of nucleotides. So these nucleotides are going to be the monomer, or smallest unit, or building blocks that make up DNA. And each nucleotide has 3 different components. There is a deoxyribose sugar, it's a 5-carbon sugar. There is a phosphate group, and then there's going to be one of 4 different nitrogen bases. We've talked about the bases before. We can have adenine, guanine, thymine, or cytosine. So one of those nitrogen bases will be attached on our nucleotide. In this picture, we have adenine. Okay, so we will talk more about each of these 3 individual components of our nucleotides very shortly, but for now, let's just talk about how these nucleotide monomers combine. So in DNA, nucleotide monomers, the building blocks, will be combined to create longer DNA polymers. So remember, poly means many. So a polymer is just a fancy way of saying that there's many nucleotides attached together. You can see that in this picture we have one nucleotide monomer here. We have our sugar, our phosphate, and our base. And then you can see that there are many more monomers attached. And as these monomers continue to attach to each other, they're ultimately going to make this long, double-stranded DNA polymer. Okay? So now let's go back and take a look at how each of these individual components of our nucleotides combine to make this specific structure. Alright, so we're going to start with the nitrogen bases. So you may remember that there are 4 different nitrogen bases in DNA. We have adenine, thymine, guanine, and cytosine. Now, as you can see in this picture, adenine and thymine are always going to bond to each other. We always have A with T. And then guanine and cytosine, G and C, are also always going to bind, and you can see that here. So adenine is always with thymine, guanine is always with cytosine, and the way that they bond is via hydrogen bonds. You can see those hydrogen bonds here. We have our hydrogen on one molecule binding to an electronegative molecule on the other. So that hydrogen bonding is going to connect our nitrogen bases. Alright, then we have two more components, the deoxyribose and phosphate, and they are going to combine to form the sugar-phosphate backbone, which creates the outside borders of our DNA. And you can see that here we have our phosphate, sugar, and they just will bond all the way down one side of the DNA, and you can see them making another backbone on the other side. And the sugar-phosphate backbone, it does act like a backbone in that it provides support and structure to our DNA molecule. It holds it all together. And then you can see that the nitrogen bases will then bond on the inside, again via their hydrogen bonds. So this is another picture of a DNA molecule. Again, we can see that sugar-phosphate backbone winding around the outside of the DNA, and they twist around each other in what we call a double helix. And then those hydrogen bonds are in the middle, so we have our nitrogen bases bonded down the center. So this is the basic structure of DNA. Okay, so let's review this really quickly. DNA, it is a nucleic acid. It is in a double helix shape, which you can see here, which just means that we have two strands that twist around a central axis. You can see the central axis here is just an imaginary line going down the center of our DNA, and then our strands of DNA are going to twist around this imaginary line, twist around each other in a double helix. And the outside of our double helix is what is called our sugar-phosphate backbone, which is just alternating phosphates and sugars connected to each other. And then the nitrogenous bases will bond on the inside of our DNA molecule following base pair rules. So adenine is always with thymine, which you can see here, and guanine is always with cytosine, and they're in the center of our ladder. Okay, so now it's your turn to do some practice. If I give you one side of a DNA strand, its sequence is A, T, C, G, A, G, C, C. What is the sequence of the complementary strand? I want you to pause the video here, grab a paper, pencil, something to write with, and try to build that complementary strand using our base pair rules. Give it a shot right now. Okay, did you do it? You should have gotten T, A, G, C, T, T, C, G, G. And the way that we figure that out, we know that adenine always bonds with thymine and guanine always binds with cytosine. So if I see an A, it has to be paired with a T. A T is paired with an A, C with a G, G with a C, A with a T, A with a T, C with a G, C and G, C and G. Okay? So, if you got this, that means that you understand how base pair rules work in DNA, which is awesome. If you struggled with this, I recommend going back in the video, listening to me talk about nitrogen base pairing again, and then come back and give this another shot. And if you are still confused, please comment in the Google Classroom, let me know so that we can go over it. Okay, so today we reviewed DNA. Where are we going next? The question I want you to consider for next time is, how do we go from this DNA that carries the instructions for life to the proteins our body needs to function? That's what we're going to be talking about more specifically in the coming videos. But for now, that is the end of video 1. You did it! Please post any questions or comments you have on this material in the Google Classroom. I would love to talk about this material with you. And also, please remember that Mrs. Liu and I are here to support you. So if we can help you in any way, please reach out and let us know. Otherwise, thank you so much for watching. Keep an eye out for the second video, part 2, coming out soon. And hope you all have a good one.